Photosynthesis in higher plants. As we all know that photosynthesis is a process used by plants and other organisms to convert light energy into chemical energy, and this energy which is released is used as a fuel for the different activities of organisms. So this light energy which is converted into chemical energy is stored in the form of carbohydrates such as sugars. These are synthesized from carbon dioxide and water. So photosynthesis is derived from Greek language, where photo means light and synthesis is putting together. See, all animals, including human beings, depend on plants for their food. Green plants synthesize their own food material, whereas other organisms depend upon the plants. So, green plants carry out photosynthesis in the presence of light. All the living forms on the earth depend on sunlight for energy. So, in the process of photosynthesis, plants use sunlight, energy from the sunlight. So, photosynthesis is important due to two reasons. One is it is the primary source of all food on the earth, and the second one is it is responsible for the release of oxygen into the atmosphere. So in this chapter we will focus on the structure of the photosynthetic machinery and the various reactions that transform light energy into chemical energy. We already told photosynthesis in the process of photosynthesis converting light energy into chemical energy. So we are going to learn this in this lesson. To know about photosynthesis, in the earlier classes you might have performed some experiments to show that chlorophyll, light and carbon dioxide are required for the photosynthesis. To know light is essential for the process of photosynthesis, you might have carried an experiment for the starch formation in the leaves. If you take a leaf and cover a partial part of the leaf with a black paper and then expose it to sunlight. After that, you see the green leaf is exposed to sunlight is turned into blue-black color, whereas the portion which is covered with black paper is in the color white. This shows that photosynthesis occurred only in the green parts of the leaves in the presence of light. In another experiment, half leaf experiment, if you take a leaf and take the half of the leaf and dip it in the solution containing potassium hydroxide, and the other part of the leaf you just leave it in the air. You just leave this for some time. After some time you will see the leaf will test positive for the starch, while the other portion that is in the tube will test negative. This shows that carbon dioxide is required for photosynthesis. The above experiments, the first one requires the presence of light, and the second experiment requires the presence of carbon dioxide. Both light and carbon dioxide are essential for the process of photosynthesis. There are some other scientists who conducted the experiments to understand the photosynthesis. John Priestley discovered oxygen in 1774. Priestley performed a series of experiments which he showed the role of air in the growth of green plants. He conducted several experiments. See, He took a burning candle and he placed a bell jar on it, and simultaneously he took a mouse and kept in a bell jar. So later on he observed that the mouse got suffocated and the candle was also off because of the lack of air. So he concluded that both the burning candle and the animal that breathed somehow damaged the air. Later on what he did is, he placed a mint plant in the same bell jar where he kept a mouse and the burning candle. Along with that, he kept a mint plant. What he observed is both mouse stayed alive and the candle also burnt. By this, Priestley hypothesized that plants restore the air whatever the animals breathe, and also the burning candles removed. That means it replaces, the plant is replacing the air which the animals are utilizing. Later on, Jan Ingenhouse performed an experiment with an aquatic plant. What he did is, the aquatic plant, he exposed it in a bright sunlight, and what he observed is small bubbles were formed around the green parts, while if he kept that green plant in dark, the bubbles were not formed. When they are exposed to bright sunlight, these aquatic plants showed bubbles, small bubbles. So later he identified that these bubbles to be those of oxygen. So he concluded that the green parts of the plants is that could release oxygen. Only green part of the plants can release oxygen. In the left side of the picture you see bubbles, and the experiment is conducted in an apparatus on the right side. Julius Van Sachs gave evidence for the production of glucose during the growth of plants. See, glucose is usually stored as starch in the green substance of the plants. Okay, the green substance we call it as chlorophyll. They are located in special bodies called chloroplasts within the plant cells. So he found that glucose is made in the green parts of the plants, and glucose is usually stored as starch, that is in chloroplasts. T.W. Engelmann considered several interesting experiments. He used a prism to split the light components, and he illuminated a green alga, that is cladophora, placed in a suspension of aerobic bacteria. In the spectrum, he observed that the bacteria accumulated mainly in the region of blue and red light. It roughly resembles the absorption spectrum of chlorophyll A and B. Cornelius van Niel, a microbiologist, based on the studies of purple and green bacteria, he demonstrated that photosynthesis is essentially a light-dependent reaction in which hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrates. In green plants, water is the hydrogen donor and is oxidized to O2. Some organisms do not release oxygen during photosynthesis. If we see some purple and green sulfur bacteria, when H2S is the hydrogen donor, the oxidation product is sulfur or sulfate depending on the organism and not the oxygen. So based on his studies, it is understood that the oxygen evolved by green plants comes from water and not from carbon dioxide. If we look at the equation, 6CO2 plus 12H2O in the presence of light and chlorophyll gives rise to C6H12O6 plus 6H2O plus 6O2. That is, the product oxygen is evolved from water and not from carbon dioxide. C6H12O6 represents glucose. The oxygen released is from water. This was proved using radioisotopic techniques. Now let's see the site of photosynthesis, where photosynthesis occurs. We know that it occurs in green leaves and in particularly in chloroplasts. If we look at the structure of chloroplast, there is an outer membrane, inner membrane, and in the chloroplast we see grana. There the thylakoids are arranged. These grana are connected by a lamella and the fluid inside is stroma. So where the light reaction takes place is in the thylakoids, that is in the grana. The membrane system is responsible for trapping light energy and also for the synthesis of ATP and NADPH. In stroma, enzymatic reactions incorporate CO2 into the plant, leading to the synthesis of sugar, which in turn forms starch. So in grana, they are directly light driven. This is light reactions. These are called light reactions which are taking place in thylakoids. In stroma, they are not directly driven by light, but they are dependent on the products of light reactions, that is ATP and NADPH. So they are called dark reactions. So light reactions takes place in thylakoids, that is in grana, and dark reactions takes place in stroma. How many pigments that are involved in photosynthesis? See, in a same plant we see different shades of green leaves. The leaves in different shades in the same plant. See, this is because of the leaf pigments that are present in the leaf. We can separate these leaf pigments by a paper chromatography. It's an experiment. You can do it in a practical session. The paper chromatography. When we separate these pigments using paper chromatography, we see chlorophyll A, chlorophyll B, xanthophylls, and carotenoids. We can identify chlorophyll A as bright or blue-green in color, whereas chlorophyll B is yellow-green in color, and xanthophylls, they are yellow in color, and carotenoids, they will be yellow or yellow to orange color. So that's why when you see a carrot, it is yellowish orange, and xanthophylls, those which are yellow leaves, they are presence of xanthophylls. So different types of pigments: chlorophyll A, chlorophyll B, xanthophylls, and carotenoids. The pigments are the substances that have an ability to absorb light at specific wavelengths. These pigments, when they absorb light, the wavelengths, if we see the wavelengths, in the blue region and the red region, there is maximum wavelength. That is, chlorophyll A is showing maximum wavelength in the regions of blue and red. That means the maximum absorption is done by chlorophyll A. So we can conclude that chlorophyll A is the chief pigment associated with photosynthesis. So chlorophyll A, even it is the major pigment responsible for trapping the light, other thylakoid pigments like chlorophyll B, xanthophylls and carotenoids, which are called accessory pigments, also absorb light and they transfer energy to chlorophyll A. These pigments also protects chlorophyll A from photooxidation. So all these pigments, chlorophyll A, chlorophyll B, xanthophylls and carotenoids, among all these, chlorophyll A is the major pigment which traps the light and plays a major role in photosynthesis.